Assalamu alaikum, dear students. Welcome to the course Principle in Animal Life 1. This is lecture number 44. The topic is Regulation of Enzyme Activity. Discussing agenda for the topic is Regulation of enzyme activity helps control metabolism. Allosteric regulation of enzymes. Allosteric activation and inhibition, feedback inhibition, and finally localization of enzymes within the cell. Lecture outcome. After watching and listening this lecture, students will know about the mechanisms employed to ensure regulation of enzyme activity. Introduction. Regulation of enzyme activity helps control metabolism. Now the cell is supplied with limited resources and it cannot afford to be extravagant. It cannot afford to use its resources unwisely. What actually happens in chemical reactions, products are produced and these products then perform functions, either they provide energy or become structural components. Now the need of the hour is to calculate which product is required, in what concentration it is required, and for how long it is required. So these three questions must be addressed. Now, chemical chaos would result if all of a cell's metabolic pathways were operating simultaneously. If all the metabolic pathways that occurs inside a living system occurs simultaneously at a time, then obviously it will be catastrophic for the cell. Intrinsic to life's processes is a cell's ability to tightly regulate its metabolic pathways by controlling when and where its various enzymes are active. We have talked about the specificity of enzymes, where the enzymes are specific to catalyze a particular reaction, but the activity of the enzymes must also be regulated and controlled so that they doesn't produce a product which is not required. It does this either by switching on and off the genes that encode specific enzymes, or as discussed here, by regulating the activity of enzymes once they are made. So this regulation can occur at the DNA level when the genes are switched off, those genes which code for the enzymes, or once the enzymes are synthesized, then these enzymes can also be regulated. They can be blocked and they can be turned into inactive state or active state depending upon the need of the cell. Allosteric regulation of enzymes. On the right side of your screen you can see a feedback inhibition pathway. This is the feedback inhibition pathway in isoleucine synthesis. Isoleucine is an amino acid, so this is the initial substrate which will enter into that pathway, and this initial substrate is threonine. Now threonine will attach with the active site of the enzyme threonine deaminase. This will form the enzyme substrate complex, and the enzyme will catalyze this reaction. Intermediate A will be produced, which will be catalyzed by enzyme 2 into intermediate B, catalyzed by enzyme 3 into intermediate C, catalyzed by enzyme 4 into intermediate D, and finally this intermediate D will be catalyzed by enzyme 5 into the final product, which is isoleucine. Now this isoleucine, which is the final product of this overall reaction, can act as an inhibitor of this pathway. This product, this final product in itself, can act as the inhibitor of this pathway depending upon its concentration and its requirement. So how this isoleucine will inhibit this pathway? It will block the enzyme by attaching to the allosteric site. Allosteric site is that site other than the active site. So when isoleucine attaches to the allosteric site, it will change the structure of the active site, and the active site will no longer be able to bind to the substrate, and therefore this pathway will be blocked. And if there is a need of the isoleucine which was produced is consumed by the cell, then there is a need to synthesize more isoleucine. And what will happen? This isoleucine, which is attached to the allosteric site of the enzyme, will be detached, and once again the active site will come into its normal shape, and it can catalyze this reaction again. In many cases, the molecules that naturally regulate enzyme activity in a cell behave something like reversible non-competitive inhibitors. Non-competitive inhibitors are those which does not compete with the substrate for the active site, but they block the enzyme activity by binding to the allosteric site, and this type of inhibition is reversible, just like the one which happened here. These regulatory molecules change an enzyme's shape and the functioning of its active site by binding to a site elsewhere on the molecule via non-covalent interactions. Now, allosteric regulation is the term used to describe any case in which a protein's function at one site is affected by the binding of a regulatory molecule to a separate site. A protein's function can be affected in two ways. First, by directly binding to the active site, and secondly by binding to the allosteric site. It may result in either inhibition or stimulation of an enzyme's activity. Now, how this process of allosteric activation and inhibition occurs? Most enzymes known to be allosterically regulated are constructed from two or more subunits. Most enzymes which are regulated allosterically are composed of two or more subunits. each composed of a polypeptide chain with its own active site. So it means it is a complex made up of several polypeptide chains, several units where each unit has its own active site. The entire complex oscillates between two different shapes, one catalytically active and the other inactive. Now these subunits combine and then form a single structure, and this single structure oscillates, adopts two different forms. One form is the inactive one and the other form is the active one. So there is a constant oscillation between these two shapes. In the simplest kind of allosteric regulation, an activating or inhibiting regulatory molecule binds to a regulatory site, sometimes called an allosteric site. So a molecule which acts as activator or inhibitor, how it carry out its role, how it inhibits or activates the enzyme. It does so by binding to the allosteric site. The binding of an activator to a regulatory site stabilizes the shape that has functional active sites. Now, as already mentioned, that this enzyme which is made up of several subunits, it oscillates between an active form and an inactive form. So when an activator arrives, it usually locks this enzyme in its active state, and when an inhibitor arrives, it locks this enzyme in its inactive state. The subunits of an allosteric enzyme fit together in such a way that a shape change in one subunit is transmitted to all others. So when the allosteric enzyme fits together in such a way that the subunits of an allosteric enzyme fits together in such a way that a shape change in one subunit is transmitted to all subunits. How? Through this interaction of subunits, a single activator or inhibitor molecule that binds to one regulatory site will affect the active sites of all subunits. So as already mentioned, that the enzyme which is allosterically regulated usually consists of two or more subunits, and each subunit has its active site. So when the activator or the inhibitor binds to the allosteric site of only one subunit, the same type of response is automatically shown by the other subunits. Fluctuating concentration of regulators can cause a sophisticated pattern of response in the activity of cellular enzymes. Now, the product of ATP hydrolysis is ADP and inorganic phosphate, and they play a complex role in balancing the flow of traffic between anabolic and catabolic pathways by their effects on enzymes. Now, ATP and ADP, they play a role as activator and inhibitor in catabolic and anabolic pathways. ATP, which is the energy currency of the cell, if ATP is in abundant amount, then it will block the enzymes which carry out catabolic pathways, because catabolic pathways quite often results in the production of ATP or energy. So when ATP is already in abundant amount and surplus amount, then there is no need to produce more energy. Therefore, it will block the enzymes which participate in the catabolic pathways. On the other hand, when ADP is in surplus amount, then there is of course a need to produce ATP. Then ADP will act as activator of the catabolic enzymes, and hence catabolic pathways will proceed. This is logical because catabolism functions in regenerating ATP. So ATP blocks catabolic pathway by blocking the enzymes responsible, and ADP, it acts as activator for the enzymes participating in catabolic pathways. If ATP production lags behind its use, ADP accumulates and activates the enzyme that speed up catabolism, producing more ATP. ATP, ADP, and other related molecules also affect key enzymes in anabolic pathways. In this way, allosteric enzymes control the rate of important reactions in both sorts of metabolic pathways. So depending upon the need of the cell, if ATP is in surplus amount, then catabolic pathways are blocked. If ADP is in higher concentration and there is a need of energy, a need of ATP, then the ADP act as activator of catabolic pathways and binds with the enzymes that are involved in catabolic pathways. Now listen to it carefully, because there is a very interesting phenomena being discussed here. In allosteric regulation, an inhibitor or activator usually binds to site other than the active site. The enzyme also has an allosteric site from where its activity is controlled. But here in this situation, a substrate binds to the active site of one of the subunits of a multi-subunit enzyme, and the response it induces is automatically exhibited by all other subunits of that multi-subunit enzyme. You can see here, this is a multi-subunit enzyme consisting of 4 subunits. Now a substrate molecule binds to the active site of one subunit, and automatically the same response is exhibited by all other subunits. The active site of all other subunits automatically comes into its correct conformation and available for binding of the substrate. So this type of activation is called cooperativity, and thereby increasing catalytic activity at other active sites. called cooperativity. This mechanism amplifies the response of enzyme to substrate. One substrate molecule attaches to one of the subunits and it acts on additional substrate molecules more easily. This multi-subunit enzyme attaches more substrate molecules easily. Cooperativity is considered allosteric regulation. Now, this is the interesting phenomena I was talking about. In allosteric regulation, usually the inhibitor or activator binds to the allosteric site. But in cooperativity, the substrate binds to the active site and the response is shown by active sites of other subunits. So you can relate this with those enzymes which are only made up of a single subunit, so as when the activator binds to their allosteric site, the effect is induced at the active site. So here in a multi-subunit enzyme, substrate molecule binds to the active site and the change in conformation of the active sites of other subunits takes place. Although hemoglobin is not an enzyme, it carries oxygen rather than catalyzing a reaction. Classic studies of hemoglobin have elucidated the principle of cooperativity. Hemoglobin has four binding sites for oxygen, and when oxygen binds to one of these sites, the other three sites automatically develops more affinity to binding oxygen. The binding of an oxygen to one binding site increases the affinity for oxygen of the remaining binding sites. Thus, where oxygen is at a high level, such as in the lungs or gills, hemoglobin's affinity for oxygen increases as more binding sites are filled, and when this hemoglobin molecule travels to areas which are deprived of oxygen, then the affinity of the binding sites of hemoglobin decreases, and thereby oxygen is released to those oxygen deficient tissues. So cooperativity works similarly in multi-subunit enzymes that have been studied. We have already discussed this pathway. Now it is feedback inhibition, in which in a multi-step biochemical pathway, one of the products act as a control, act as a regulator on the pathway. So this is the pathway of the synthesis of isoleucine from threonine, and right at the start, threonine deaminase converts threonine into intermediate A, intermediate B, C, and D, and each step is catalyzed by different enzymes, and the final product isoleucine is synthesized. So when isoleucine is in surplus amount, in abundant amount, then this isoleucine itself acts as a negative control on this pathway, and it attaches to the enzyme's allosteric site, and thereby the enzyme's active site is no longer able to bind with threonine. When isoleucine is consumed and it is in deficient amount, then this association is loosened, isoleucine is released from the allosteric site, the active site comes into its correct conformation and available for the binding of threonine. So this is feedback inhibition, in which a metabolic pathway is halted by the inhibitory binding of its end product to an enzyme that acts early in the pathway. So this enzyme is acting early in the pathway. Isoleucine is the end product, and now it is acting here as an inhibitor. Some cells use this 5-step pathway to synthesize the amino acid isoleucine from threonine, which is another amino acid. So when isoleucine accumulates, it slows down its own synthesis allosterically, inhibiting the enzyme for the first step of the pathway. Feedback inhibition thereby prevents the cell from making more isoleucine than is necessary, and thus wasting chemical resources. Localization of enzymes within the cell. Now the cell is like a container in which different chemical reactions are going on. It's a chemical container, but these chemical reactions are not going haphazardly. There is a proper control, a proper regulation, a proper check and balance, and this is important for the homeostasis of the cell and as a whole the organism. So some of the enzymes are localized, they are located in specific compartments of the cell. For example, in endoplasmic reticulum we discussed molecular chaperones which are present in its lumina. In the lysosomes, hydrolytic enzymes are present. In the mitochondria, the enzymes which carry out electron transport chain and Krebs cycle are present. So the enzymes are localized in this way, and also sometimes the enzymes which catalyze different steps of a single metabolic pathway, those enzymes aggregate and form a multi-enzyme complex. Suppose a biochemical pathway is catalyzed by 10 enzymes. Now these 10 enzymes, they aggregate together, they accumulate together and form a multi-enzyme complex which catalyze different steps of this biochemical pathway. Here you can see in the membrane different enzymes are embedded, and they usually catalyze different steps of a biochemical pathway. The product of one reaction is a substrate for another enzyme, and the product is a substrate for yet another enzyme, and similarly different steps of a reaction are catalyzed in a proper manner. Another example of multi-enzyme complex is pyruvate dehydrogenase. This enzyme converts pyruvic acid, the end product of glycolysis, to acetyl coenzyme A, which is the starting point of Krebs cycle. Now this is some more information on the localization of enzymes, where the enzymes are localized to specific compartments within the cell. In the coming chapter, we will discuss aerobic cellular respiration and its various steps. This was the end of the lecture. Thank you.